Classes of proteins that mediate facilitated diffusion. Carrier protein mediated. Carrier proteins called transporters or permeases non-covalently bind specific molecules to be transported on one side of the membrane. They then undergo conformational changes that allow the molecule to pass through the membrane and be released on the other side. The classic example is the movement of glucose mediated by carrier protein, glucose transporter, GLUT. There are various isoforms of the glucose transporter GLUT. In human, there are 14 GLUT proteins which possesses various substrate specificities and involve in the transport of glucose and other hexose. The chloride bicarbonate exchanger, also called anion exchange protein, present on plasma membrane of RBC, mediates the transport of two anions simultaneously. Bicarbonate ion moves in one direction and chloride ions moves in the opposite direction. The exchange is electroneutral because of no net transfer of charge. Channel proteins mediated diffusion. Channel proteins form open pores through the membrane, allowing the free diffusion of any molecule of the appropriate size and charge. Rate of diffusion mediated by channel protein is higher than those mediated by carriers. Channels typically show less stereospecificity than carriers and are usually non-saturable. Channel proteins concerned specifically with inorganic ions transport are called ion channels. Ion channels are highly selective. Most of the ion channels are not permanently open. Some channels, called voltage gated channels, open in response to change in electrical potentials. Others, called ligand gated channels, opens in response to the binding of ligands. Many animal and plant cells contain specialized water channel in their plasma membrane to facilitate the water flow called aquaporins. These are a family of transport proteins that allow water and a few other small uncharged molecules such as glycerol, ammonia to cross membrane. Aquaporins AQPs assemble as homotetramers in which each monomer consists of 6 membranes spanning a helical segments with cytoplasmically oriented amino acid and a carboxyl termini. Two hydrophobic loops contain conserved asparagine, proline, alanine, which is NPA motif for selectivity of the channel. Each monomer functions as an independent pore.